So we're going to get started, I guess, in about 2 minutes or maybe a little less.
Okay, Bruce, will my screen come right back up, or do I have to link back in and share?
You'll need to share.
Okay,
So
got it.
if you go share application, then it should, and then you should be able to pull up directly the presentation.
Okay.
Get started here in 30 seconds. All right, let's go get started. So I'd like to welcome everybody today to the Plant and Plant Microbiomes learning sessions, and just give a little introduction to the overall sessions that we're doing here. These learning sessions have been put together by myself, Deborah Wells, and Charles Quincy, and this program has been funded by the NASA Space Biology. Hang on a second. So what these sessions are really about is about a huge challenge that's facing NASA as we move out to the moon and to Mars. And it's about where we'll be tomorrow, and it's really about food. And because the astronauts are going to have to be far more sustainable in the future than they ever have had to been in the past. And this is such a big problem that NASA's tackling it in 2 different ways, or more than one way anyway. One is they're actually doing a centennial challenge where teams will compete to design sustainable food systems for astronauts, and that'll be released, I believe, in January of 21. And the other is really to try and understand the impact of these deep space environments on the biology so that we can design better hardware and do better job of growing crops in space. And that's really what these sessions are about. So what do those environments look like that we'll be moving into? Well, as you know, NASA has been directed to have people back on the surface of the moon in 2024 as part of the Artemis missions. And as part of that, there are 2 space vehicles that are currently deep into development. One is the Orion space vehicle shown down here, and its job is really to ferry astronauts from the Earth to the vicinity of the moon, where it can then dock with this vehicle, which is the Gateway vehicle. The Gateway can serve both as a gateway down to the surface of the moon and as a science laboratory in orbit of the moon. Of course, to get down to the surface, first of all, I guess where we want to go is to the South Lunar Pole. This is Shackleton Crater, which has these eternally sunlit regions, which are good for generating solar power, and these eternally shadowed regions, which are supposedly rich in water. Get down there, there are 3 landers under development that are on the drawing boards. Only one will eventually be chosen for the job. The first is this Blue Origin lander, which is reminiscent of the original Apollo landers, 2 pieces, a descent stage and an ascent stage. And that's being pioneered by Blue Origin and the national team, which is made up of Northrop Grumman, Draper, and Lockheed Martin. The next one is the Dynetics Human Landing System. It's different than the other system in that the vehicle both descends to the surface and the entire vehicle ascends back from the surface at the end of the landing. And the third, of course, is the SpaceX Starship design, which is probably the biggest of the three. And these are all very different vehicles and will offer very different opportunities for doing science on the moon. So what do we need to know to get to where we want to be? Well, to know that, we need to know what our knowledge gaps are, and we need to know how to fill those gaps. And to do that, that's a lot of what we're doing in these meetings right now, is trying to assemble a diverse community that can help address those issues. And what that community looks like is it's a community of space scientists, plant microbiome scientists, space platform designers and builders, science hardware designers and builders, people who know a lot about the space environments, and then also about the cutting edge science tools. Because if you think about, I guess what I mean by that is if you think about sequencing for a moment, it wasn't very many years ago, it took millions of dollars and over a year to do a single human genome. Now that work can be done in about 4 hours over a cup of coffee for just a few hundreds of dollars. These sessions have been designed to try and get everybody onto the same page in that community. So really we're encouraging the engineers to attend the science talks and the scientists to attend engineering-oriented talks, because we want everybody to understand the problems that are facing each one of those groups. It's only in that way that we can design the best hardware and the best science to meet the challenges that are in front of us. And ultimately, we'll have some workshops at the end of this. The first of those will be just after the ASGSR meeting in November. And there we'll be addressing some of they're trying to come up with a lot of science for design science white papers that will be fed into the National Academy decadal plan. And then another one will be done in probably January. We'll be looking more at transformational hardware to help the science get done in those different locations. But right now, it's really about generating ideas and white papers to feed the National Academy's decadal. So with that, I'd like to turn it over to Christina Khodadad. Actually, we have two speakers today, Christina Khodadad and Ani Dixit, and they'll be speaking to us about the plant microbiome. So Christina will be speaking first. Go ahead and take it away. So, all right, we can see the slides. Just go to presentation mode, Christina, and then we're good.
Okay. I am in presentation mode. Does it not appear that way?
Yeah, I think go ahead.
Okay. Well, I wanted to thank you all for asking us to participate in this and provide some of our information on the plant microbiome. So to introduce the plant microbiome, we really would like to just basically define it. And there are so many different definitions for the microbiome, and some of it depends on your focus point, whether it might be ecological or host-dependent, genomic dependent, or even a combination of those. So the plant microbiome, for what we're going to work with, is defined as the distinct microbial communities that are associated with the plant host tissues. And it also includes the area that surrounds those tissues. Now, these could be well-defined habitats, and many times they have very distinct physiochemical properties. So the plant microbiome consists of numerous microbes. We think of microbes, we always think of bacteria, but these can include bacteria, fungi, viruses, algae, archaea, protists, and others. So it is a very large community. We can
So, Tina, can you move a little closer to your microphone?
I'm sure. I'm sorry, I'm kind of froggy today too.
All right. Sorry.
The microbiome can be subdivided into categories, where it's anything as simple as the phyllosphere, which is all the tissues that are above ground, the rhizosphere, everything below ground, and it's all encompassing there. With the endosphere, incorporating everything that's internal. But plants are not really homogeneous habitats. Therefore, many times we'll even subdivide that. So we can subdivide it based on the plant organs or different plant tissues, such as leaf, stem, fruit. So again, you can set up your microbiome based on many definitions. Microbes are the key players for plant health and productivity, and they will change. Those communities will change under certain circumstances. So, such as pathogen presence, or even a change in
So
environmental
Tina,
conditions.
Tina, I'm sorry to
Yes.
interrupt. Are you changing slides?
No, not yet.
Oh, okay.
Yeah, I
Thanks.
did.
Oh,
Okay, I did, but
we're still on slide 1.
okay, there we go.
There we go. Thank you.
I'm seeing it on mine, but not on yours. Okay, sorry. Um, with what we know about, made attempts to manipulate that so we can provide some additional plant benefits. Through the manipulation, we can reduce. We think we can reduce the incidence of plant disease. They've been using it to increase agricultural production. And reach more sustainable practices. We've even been using it to reduce possible chemical inputs such as pesticides. So there's many different things that the microbiome can do for us and for the plants. So the functions of the microbiomes, or all the plant-associated microbiomes, are to promote growth and acquire nutrients for the plants, protect and defend the plants, as well as provide stimulants which are helpful in stress control. So these benefits can be direct or indirect, such as what we see with the nitrogen fixation in plant growth promotion, ammonification via the mycorrhizal fungi, and then also acquiring elements, essential elements for the plant, such as potassium or iron. Also in the defense, the microbiome can help protect against pathogens, provide disease resistance. Plant roots can stimulate and support these microbes, and then help protect the plant itself from pathogens or other pests. And as well can help to unlock access to essential nutrients while it's doing that. The microbiome does help to produce the stimulants, again, that will help with stress control. An example is plant hormones, such as stimulating growth by auxins and gibberellins, cytokinins, also, it provides an ACC enzyme, which is can provide which is a precursor, of course, to ethylene. And these are things that will help the plant in maintain its health and growth. So the bottom line really is for all of this is that the plant microbiome interactions are going to improve growth and health and of all the host plants. There are numerous things that are going to affect the microbiome, both biotic and abiotic. We have, we see general trends across the board with when we characterize the phylosphere and the rhizosphere, as well as the endophytes in those communities. Endophyte diversity is pretty dynamic, especially in the rhizosphere. And it's influenced again by both biotic and abiotic conditions. The phylosphere is generally a nutrient poor environment. There's usually low diversity of microbes there, and they are, the plant and the microbes are subject to environmental conditions above ground, such as temperature changes or radiation, moisture. And at times, even the plant genome itself will have an influence on the phylosphere and the leaf endophytes. In the rhizosphere, again, there are numerous biotic and abiotic influences in soil or soilless conditions. We see the influence on the root structure, and the root structure is in direct contact with the microbiome. There are plant microbe interactions, and again, as the conditions change, they will change as well, and even microbe to microbe interactions. Then you put all that together, in these two areas, the rhizosphere and the phyllosphere, are crosstalk between the two, and this in turn can provide plant growth promotion via phytohormones, increasing access to major nutrients, and also provide some biocontrol. And this will help, again, promote plant growth through protection of plant pathogens. So again, the microbiome here is intricately linked to the host, and it can it be influenced by the host species itself. Those that come from produced by root exudates or influenced by the root exudates. It could be influenced by the host's life stage, or even the life's health itself, the host itself. So an example of those influences is just to, in this slide, just really shows some of the selection pressures to the microbiome, in particular the core microbiome. Ecological factors are going to help shape the diversity and the composition of the microbiome. This is just an example of how an abiotic factor, such as temperature, due to maybe a change in location, is going to have an influence. Each area will, or each plant will have its core microbiome members, which will be influenced. Usually they're unique to the species or the cultivar, so over time they're going to be influenced by the host itself and other plants in the area. Geographic location, which again is going to change many of the abiotic factors such as temperature. They'll also see changes in the microbe to microbe interactions because the microbe community is changing. And with all of these types of changes and things to consider, it will really propose a challenge on how do we study these things? How do we study these communities? And their responses to the plant microbe and microbe to microbe interaction. So there are numerous methodologies out there that are usually done in combination. This is one of the ones that I've noticed that I came across just recently by Berg. It's 2020, where they've started to look at cell sorting platforms, spectroscopy, and then leading into the omics, which is becoming more and more prevalent and useful in studying microbiomes. The key to know, the key is really knowing who is there. And that can be done either through culturomics and microscopy, which is the most basic method, and then moving on to metagenomics. With DNA, we can look at who is there based on their genome, either using markers such as ITS or 16S or whole genome. And that gives us an indirect method of looking at their metabolic potential. What can they actually do in these microbiomes? But looking at the, taking that even further to the RNA and proteins and their metabolites, we can actually see what they are doing in the microbiome. That gives us an idea of whether they're going to be beneficial to the plant and how they are benefiting the plants. So that's just one of the methods. A second method is, again, trying to combine the omics, in this case, metagenomics and metabolomics. What they've done here is they've looked at healthy tissue and stress tissue. And by looking at the microbiome and seeing who's there in, say, the leaf or the root, via the DNA and the metabolites, they can determine indirectly who might be key players in this process. By knowing the difference on where are the changes with the unhealthy tissue, they can look at the microbes, move on to a culture-based method or in vitro testing to see what the interaction actually might be, if there is one. Through that combined effort, we can then look at the whole genome sequencing and build a consortia of microbes that might be beneficial to the plant. As I said, might be, so that might lead then to an in-plant test or an in vivo test and look at the plant phenotyping, what is happening when these microbes, this microbial consortia is added, and then find out what the molecules are actually providing that benefit the host. And that brings us back circle to using the metagenomics and the proteomics or protein work and the metatranscriptome. So these are just two variations, but they all use the same type of testing processes and protocols. So those are the processes that we have also used on some of the samples that we have brought back from the ISS. NASA's been growing plants on ISS for a number of years, and we've been looking at the microbiomes of some of the tissues that's been returned to ground. There are two plant growth facilities on ISS. We heard about them last week from Howard. The veggie unit and the APH, or advanced plant habitat. We've worked primarily with veggies, so that's where I'm going to concentrate my efforts today. Both are enclosed growth spaces, but they do get exposed to station environmental parameters. And so we can look at the variation of what we're seeing on station based on and compare it to what we see on ground. We also run ground parallel studies of with the same type of conditions as we see on ISS, with, of course, the exception of microgravity. Knowledge that we've gotten from these really coincides with what general knowledge is in the field of plant microbiomes. We know that both terrestrial and aquatic systems, and by aquatic I mean really soilless systems, they all require microbes. Microbes are important to the plant growth, and again, it provides growth and health and productivity to the plant. Plants will actively recruit the microbes in order to boost their nutrients and help them with nutrient availability. Microbes are going to be supplied by the surrounding environment, whether it comes from the atmosphere, the water, the watering system, and so the plants will make use of those. They will also recruit them based on whether they have to fight off pathogens and also reduce any stress responses. And we see some of that occurring as well. And then hydroponic systems, we have found seem to be more favorable to bacteria than fungi, and some of the ground systems have determined that, ground testing has determined that as well. So we've put a lot of into looking at the bacteria and fungi too of the microbiomes in veggie grown lettuce. And when we look at this at a higher taxonomic level, we find that at these upper taxon levels, such as class and phylum, we see the same trends as we would in any other ground study. We see high levels of proteobacteria. These are prominent, and alpha and beta proteobacteria are usually found in the highest levels. That we see in as well as in all ground systems. But as we drill down into the lower taxonomic levels, we can really see and pinpoint a little bit closer who is really there. We can take it to the genus and species level, depending on your bioinformatics pipeline. And we can look at both plant, phylosphere, and root tissue, as we did here. Here on the left, we have our plant tissues grown in flight and ground. And the same over here, we have our root tissue, flight and ground. So you can do lots of different comparisons. But what we see here overall is that we have higher diversity in our root tissue. That's not unexpected. We see some overlap between the phylosphere and the rhizosphere. Again, that's not unexpected. So there are lots of similarities with terrestrial systems. But now we know who's actually there and will they be beneficial, but that's going to require additional testing. We know who's there. So we did a further study on ground where we looked at core microbiomes. These are those that are usually unique, but are there in high numbers as well. We looked at the core microbiomes for red romaine lettuce and Mizuna mustard, which is another crop that's been grown on ISS. And we can see that there is some overlap. They share some of the same species, but there are some that are very unique as well. And so like in Mizuna, we see Azospirillum and Bacillus. These have been previously identified as key players. In red romaine lettuce, we see Bradyrhizobium and Burkholderia. Again, these have been key players identified via terrestrial systems. So those might be a good place to start. We've also looked at comparing our seed treatments. Sorry. In our seed treatments, we usually, when we send seeds to station, we have to sanitize the seeds. Well, that is one way of manipulating the microbiome because we're removing the microbes that are on the outer surface of the seed. So we wanted to take a look at that. So again, this is just one manipulation. Comparing these seed treatments, we can get a clearer picture of similarities and differences if we are removing good microbes or bad microbes, and are we really, which ones are being shared, and that helps us again identify the core. From these two studies, it has provided us with the information of who's there, but we still need to take it another step further. What are they doing? How are they contributing to the host? And so are they being beneficial or not? So we took it to the next omics level, and that's the metatranscriptome, where we looked at RNA. These are the genes that are being expressed by the microbes that are there. And from this, we can look at, it provides us with different types of information. One, it does, from the genes that we are able to identify, we can get a look at who's there. Again, it's a confirmation of what we've learned from the genome, the DNA work. But it also gives us an idea where we can look at it via plant age, or we can look at it in plant part. In this particular example, I'm using root tissue, and it gives us over time our sanitized and unsanitized seeds. And we see that there are some trends here that we can investigate right at the get-go. We see that in early day 7 samples for unsanitized seeds, we see that certain microbes are not present, whereas the sanitized seeds, we do see certain microbes at this point in time being present, possibly recruited. It's something we don't know yet. And then again, we see a similar trend in this series of organisms. But in addition to knowing who's there, we can look at their functional groups and it gives us an idea of what they are doing. Now, we still need to take this another step to try and link this side with this side. And that's basically a software issue. And we will use additional pipelines in order to determine that. But this is, again, a good indication that certain things are going on in upregulated or downregulated modes. So, that's what we've done for our soilless system on veggie and here on ground in different tests. But there are other types of systems that are available that may need further indications of what they can provide. Aeroponics is a system that has potential. Usually when water or space is in a limited quantity. But when we looked at this data, we also saw similar trends of what we saw in our soilless systems and in the terrestrial systems. The things that stood out to me was again the increase in proteobacteria. And so we know that proteobacteria are probably going to be key players and may require additional investigation. There's also cyanobacteria here, and cyanobacteria have also been known to promote plant health. And so there's the things that we need to investigate further, but we're seeing trends on different systems. But what does that help us do when we want to go to Moon and Mars? The best way to what is going to be the best way to proceed with lunar and Martian agriculture? These also is going to be a challenge. We know from some of the studies that have been ongoing for numerous years that we can supply what the plants need via fertilizer when we use soilless systems, but there are other things that need to be considered, and there are still deficiencies. So what do we add to that? And so a lot more work needs to be done with regolith, and we're finding more and more studies out there now that do help with that. This particular regolith study was done in 2013, 2014, and they looked at multiple types of plants and which ones that actually grow and which ones survive. They looked at numerous things, but I just wanted to, who was going to survive this? Some of these crop plants are ones that we utilize and grow on station, lettuce and tomato in the future. And we found that they grew very well in Martian regolith and well on Earth systems, but the lunar regolith that they used was not so helpful. So again, more studies might be required. We heard from one just the other day, Dr. Rafael Luyero up at Winston-Salem State University is also doing some studies with regolith, and he may be able to shed some more light on what is yet to come for lunar and Martian agriculture. Okay, so there's still a lot of work that needs to be done. What can we do to help these systems along, help promote plant growth? And what we see is that there are screening methods available with the microbes that we've identified, which ones are good, which ones are bad, which ones do we need to test further. These are just assay screenings that we've started at KSC, and they might help us to identify some of these plant growth promoting bacteria. And with that, I think I will be turning this over to Ani to tell us a little bit more on how we will identify these and what we go from there.
Thanks, Tina. Turning it over to Ani, I'll just mention that if you have questions, please put those into the Q&A panel. There are too many people on the phone for us to unmute all your microphones. Thanks. Go ahead, Ani.
Thank you, Tina. Really appreciate you handing me over this. So I'll carry forward from here. So while we are on this subject, I would like to mention that uh, the plant growth promoting bacteria, as Tina was mentioning, play quite a few different roles in how it can help plants do better in different environments. So what I'm going to share further here is look at some of the key considerations that one need to take into account if you're going to use these bacteria for plant growth promotion. And so what are some of the key considerations for that? Thinking from, so this particular paper came out in 2017. So I like this one. It's just a mini review. And it talks about what you need to do or in order to exploit some of the plant beneficial microbes. And it talks about some of the key considerations. And one of the key consideration is we have to screen a large number of isolates. I mean, people who work in this field, they know that no two rhizospheric soil conditions are similar, and therefore the plants that grow in those conditions, their associated microbiomes are also quite different. So despite of having a large variety or diversity of soil microbes, plants are known to be very specific, or they have a selective microbial niche. They recruit very specific things for their own benefit. Now, thinking from the metagenomic dataset analysis point of view, these different rhizosphere may exhibit very little overlap in terms of plant enriched functions. So I guess if you are planning on doing or working on a beneficial microbe strategy, then the first thing would be you will be end up screening a large number of isolates. The second consideration would be an ecological one, where you have to look at the plant beneficial function of the microbes itself. And what I mean by that is the plant growth promoting bacteria or the candidate that you will be introducing into the soil, it has to be able to invade into the native microbiome that's associated with roots or phyllosphere, as Tina was mentioning in some of the previous slides. Now, ability to invade is one thing, but be able to persist in those environments is quite a bit of challenge for that good bacteria that we're introducing. Also, the environmental or abiotic conditions or the biogeographic condition in which the plant is growing also plays a critical role to the success or failure of the plant growth promoting activity. And nonetheless, the plant microbe interaction in the soil-like environment itself. So there are quite a few different parameters that are playing simultaneously to decide whether the plant growth promoting bacteria is going to be a failure or a success. Also, the third consideration would be at the diversity of the inoculum itself, whether you inoculate a single bacterium or a mixture of multiple bacteria. So this is always a subject of debate, and more and more literature is coming up that showcases that a complex inoculum in most of the cases is a better option to go about, because you can inoculate bacteria with different functions, so they'll be able to compete or they'll be able to persist in that native microbiome quite efficiently. And the last but not the least consideration would be the genetic control of beneficial plant microbe interaction. So the host genetics, so in this case, the plant genetics itself plays a bigger role in whether the plant growth promoting bacteria or the good bacteria is able to survive in this kind of a situation or not. There have been reports that talk about the microbiome changes between even the similar different accessions of the same plant. So that's why the first point that the plants are highly selective niche, microbial niche, kind of stays very true there. So with biogeographic location, the accessions, the microbiomes are constantly changing. Then how does one go about constructing a synthetic microbiome or a synthetic microbial consortium? This is one of the flow diagrams I'm adapting from this paper that came in Frontiers in Plant Sciences in 2018. And just to go over the figure, they recommend you start, and this is one of the ways you can do this. So this is by no means an all-comprehensive approach of constructing a synthetic microbiome or a consortium of that. So you always start with good quality crops because then you can assume that there are good bacteria associated with it. Then based on the isolation of the core microbiome from roots, we can basically do a high throughput sequencing, the metagenomic analysis, and figure out what is the core microbiome of this particular plant and what are their hubs. Once you have that information, then we go to something called, they recommend going to this Komodo. Komodo is basically a database that deposits or that has a repository of all the known media combinations that works for bacterial growth. So based on that information, you can select what media will be appropriate for the core microbiome that you just isolated from these good or high quality crops. But then comes the real work, because you're not only once you start culturing these microbes in the laboratory condition, then what you'll be looking for is the microbial synergism as to whether they like each other, they hate each other, they don't get along with each other. But then whether they are able to provide the plant growth promoting activity is the key question that you'll be looking for. And of course, when you have this mixture introduced to some other plant with its own core microbiome, how that dynamics is going to change. Once you have all of these factors under control or somewhat understood, then you can really go for the actual culturing of the synthetic microbial consortium, add that as a biofertilizer, and then on to the fields. Once it is in fields and applied to the plant, then come the actual evaluation in terms of efficacy and the ecology of how that played out in the whole scenario. Then some of the parameters that you'll be looking for is growth, mineral nutrients, metabolites, or the function or invasiveness or persistence of this good synthetic microbial consortium that you just created. Now, whether this is going to be successful, it's a whole different question at this point, because it needs studies through generations as to whether this is able to persist, able to withstand the environmental changes and other parameters. So this is more of a general idea how one can go about constructing a synthetic microbial consortium or a community. But then the next question is, how do you introduce that? So this is one of my favorite papers. I think this came up in 2017. So when you have uh, that good synthetic microbial consortium or community you have, how can you introduce to the plant? So in this particular paper, they did something really nice. So in this one, they are introducing this Paraburkholderia phytoferment strain, and they inoculated or they sprayed straight onto the flowers or the maize kernels in this case. So when you spray that bacteria, the bacteria tries to integrate itself into the developing seeds. And when you get the seeds, the next generation is supposed to have these good bugs, not only in endosphere, but also in the rhizosphere. So this is a phenomenal strategy. So if it is working for a single bacterium, it might even be feasible to try this on a synthetic microbial consortium. Now, what I'm showing you down here is this is how they verified that the paraburkholderia is in fact integrated into the seed. So what you're looking in the panel E is a cross section of the maize seed, the developed or the mature seed, and you can see the endosperm followed by this embryo. Now, this blue color on the embryo is coming from this bacteria itself, because the bacteria is tagged with an enzyme called GUS, glucuronidase synthase. So when you provide it an appropriate substrate to that, the enzyme reacts with it and provides blue color. So you can see that the bacteria is not only able to stay in the seed, but they have integrated themselves into the embryo. And not only embryo, when the seed germinates, you can see those bacteria kind of lightly in the radical as well. So this strategy definitely works, but whether this will work for a synthetic microbial consortium, it's a different question, but it's a nice way to look at this. Being said that, let's switch the gears here a little bit and let's talk about we just talked about the good bacteria and how they can be integrated into the synthetic microbial consortium and what sort of things they can do for plant growth promotion. But the other key question is, how does the plant microbiome change? Um, under pathogen attack. And what I'm showing you here are a couple of pictures. One of the picture is a tomato that is infected with Pseudomonas syringae, and the picture below that is of an orange that is infected with a fungus called Elsinoe australis. So yes, everybody knows plant pathology wise, yes, we have seen infection on fruits and other things too. But before we go into the details of what really happens to the plant microbiome under pathogen attack, I want to introduce this a fundamental concept in plant pathology. It's kind of plant pathology 101. This is something called as a plant disease triangle. And the gist of this figure is you have to have all of these three parameters, the environment, the pathogen, and a susceptible host present in that environment for the manifestation of the disease itself. If any of these parameters is missing, then the disease manifestation doesn't happen. So it's a quite a fundamental concept to remember that you have to have all of this for the pathogen to attack a susceptible host. Now, looking at some of the details as to what really happens under a pathogen attack, um, this is one of the reviews that came in annual reviews in phytopathology, and I really like this figure. The reason is that because it explains what really goes on, at least in some terms. Um, so what you're looking at in this figure is this is the plant stem, the outer side, the inner side, and you're seeing a lot of bacteria, or which are technically endophytes. Now, this red blob is our pathogen that is attacking the plant. But so some of the key consideration or some of the key aspects of this figure is what really the pathogen is doing. So pathogen is trying to form different things like polysaccharides, phytohormones, virulence factors, and it's when it is trying to suppress the plant's defense responses. But in return, the plant is also producing reactive oxygen species, defensins, and other things to counter attack that thing. But then look at this big green arrow and the purple arrow that's going back and forth. So the endophytes that are present in the stem, as you can see, they are interacting with each other, and they're just not interacting with each other. They're communicating each other through quorum sensing. They're exchanging their genetic material, so they can put up a fight against this invading pathogen. So if you look at this picture as a whole, it appears as a very coordinated attack by the plant and its endophyte against this invading pathogen. So this is a very simplistic approach of looking at things, but things in reality are way more complex. Speaking of how does the plant is responding to this pathogen attack, I want to focus on this particular box for my next slide because it's looking at the MABs, which is also microbe-associated molecular patterns, and the triggered immunity from those specific aspects. So briefly, the microbe-associated molecular patterns are kind of signals or certain structures on the pathogen that plant can recognize, and then the plant can decide whether to attack this thing because it recognizes those structures. Okay, moving forward. So microbiome plays a very essential role in this immunity or the innate immunity of the plant itself. So what I'm showing you here is there are two different parts of the immunity or the innate immunity to plant. One of them is a pathogen recognition receptors triggered immunity, which is the PRR immunity, and the other one is the nucleotide binding domain. These are part of the innate immunity of the plant. But when you introduce the microbiome into the whole mixture, that's when the microbes are not only helping plant defend itself against the invading pathogen, but also contributing some of the key aspects or increasing the plant's immunity towards the pathogen, and that's what we called as a direct a microbiota modulated immunity or a direct microbial competition, as we just saw in the previous figure. So keeping all of these things into consideration or making all of them mesh together, one of the key aspect when you are thinking about a synthetic microbial community design is to have some sort of a predictable outcome of the beneficial effect, whether that's plant growth promotion or be able to defend the plant against invading pathogen. And how you can do that is by increasing the robustness of beneficial microbial traits by expression of the microbes by increasing the redundancy in functional aspect and thus increasing their dominance. And what I mean by that is if you have a microbial community and one of the member gets taken out or gets dominated by the core microbiome, then it's always have a good idea to have redundant microbial functions just to keep take over that. So that brings me to my next point. So these synthetic microbial communities can also act as biostimulants. Sorry, before I go to that, I'm just want to give you a quick example as to how plants can recruit specific bacteria when they're under pathogen attack. So what I'm showing you in these plate pictures are three bacteria. So this is the case in Arabidopsis thaliana, and which is under attack by this fungus or pathogen. This is called Hyaloparanospora arabidopsidis. And when Arabidopsis is under attack by this pathogen, it selectively recruits Xanthomonas, Stenotrophomonas, and Microbacterium. When they are grown independently, they're just fine. But when you grow them close to each other, they somehow form that biofilm. And as you can see, they are already interacting with each other. And their interaction is key for Arabidopsis to fight against Hyaloparanospora. This is phenomenal. I mean, this is the real example of how plants can specifically recruit certain bacteria from soil to protect itself. And in the table, I'm just giving you more examples like Fusarium oxysporum, what are the effects, and what particular plants. But then this particular example of arugula at the right-hand side bottom of the screen, you can see that the arugula plant is loaded with this Pythium fungus. So it's obviously there are quite a bit of stress symptoms going on this one. And as soon as you introduce the Pseudomonas or Bacillus subtilis, it's looking like it's able to keep the Pythium down and still able to help plant do better in terms of growth and other things. So again, so coming back to my point a minute ago that this synthetic microbial consortium, carefully designed, of course, can be used as a biostimulants. So biostimulants can be an approach to synthetic microbiome, as I was saying earlier, a minute ago. So what are really biostimulants? So EPA describes biostimulants as something that when applied to seeds, plants, has a beneficial effect on the plant or the crop yield overall. Now, what constitutes biostimulants? So biostimulants can be a lot of different things. That's why the definition is kind of loose. So it can be acids, microbials, or the microbes themselves, or the microbial byproducts. So there are quite a few things that are constituted as biostimulants. And biostimulants act in different ways. So they can help plant increase their metabolism, improve the crop yield, or reduce the effect of biotic stresses. One such example of a biostimulant is a microbial biostimulant. So in this case, what I'm showing you, these lower two pictures, so this particular picture of Mizuna growing in these containers is being treated with a fungus called Cladosporium spherospermum, and the strain number is TCO9. This work was done by the USDA Agricultural Services Lab, Research Services Labs in Carnesville, and we got the strain from them, and we are working with them. So what it showcases is that TCO9 is able to promote growth of any plant that it comes in contact with. So this is true for the Mizuna test that we are doing, but it also works. So this is straight from their publication that came in Produced in Plant Sciences last year. So they are showing the cayenne pepper, and you can see that after treatment, the plants are quite big. Also, there is quite a bit of root mass. But I want to draw your attention straight to this very last panel here for the yield. So it's not only increasing biomass, but it tremendously increases the overall food production also. So this is a real biostimulant. So it's not affecting the plant anyway, but it's affecting it in a good way and increasing the total biomass as well as the yield. With that, I come to my summary slide, and just to put all of this into a few sentences, that microbiome interactions are quite intricate and they're exclusive to the host. And of course, the stability of a given microbiome or rhizospheric microbiome depends upon the plant type. First of all, the soil is of course important, or the things that we do in artificial media composition also for the application that we are looking into. Knowing what is the composition of the microbiome is great, but it'll be much more worthwhile to know what they were doing at that present space or time. And of course, synthetic microbial communities, if carefully designed, they can act as biostimulants. They can not only act as growth promoters, but they can help plant alleviate some of the abiotic and biotic stresses. With that, I would like to thank for the opportunity, and we can try to answer some of the questions. Thank you.
Thanks, Ani. Thanks, Tina. So there are lots of questions people have been putting into the Q&A. So we don't have a lot of time left, so we won't be able to answer all of them, but we will try and answer those and reply to you after the conference is over. I would like to take a couple of questions, though, and one that came up, I think early on, was, is there a known consortium of basically beneficial microbes that can be used to optimize growth for the plant? So this is kind of, to me, the panacea formula. Is they're a mix of stuff that we can put in that we know always works, and who should be in that formula?
So again, I mean, most of the things that come up as so you must be getting some of these beneficial microbiome as or a mixture of microbes from outside, like a commercial vendor for that matter. But it might work for certain specific plants, but will that work for every single plant? I'm not so sure about it. And again, as I said, the environment and the soil type also makes a lot of difference, whether that concoction of microbes will be able to work in those situations. I don't know, Tina, if you have anything to add.
Well, the other thing that I've seen is mostly these are commercial products. And again, they're going to work for certain plants. They're going to work in certain environments, and scale is a major consideration. What might work in a small plant habitat would not work in an agricultural field. And as far as I know, a lot of the work that I've encountered has been for agricultural field work. They may work for a very short time, but again, they don't last.
And I'll just add one comment to that, because I do have some experience with biologics from my background in crop science. And what's well known in that area is, as a problem for those products, is they have to be handled very carefully, because essentially they're living products, right? They're actually biologic. Right. They can't be in a farmer's shed on a hot day in Iowa where the temperatures may exceed 110 degrees for long periods of time. They don't last well under those conditions. So there are some products, but I also support what you, Tina, and Lonnie said, is, these things also tend to be a bit specific for soil types and definitely for plant types. So another question is our ultimate goal to 100% characterize synthetic plant microbe for all plants in space habitats? Is there any room for investigating the possibility of inoculating with more complex, undefined communities, say from real Earth soil? Or a simpler question might be, what's your real goal here?
Finding the good in the bad. A lot of what we've done was started out based on to maintain food safety. And so that led us to, and that's always going to be a consideration. So when we've, in looking at the microbiome, again, that is also a way to maintain food safety, but also to provide a better yield, a food resource for the astronauts. But once you can run the microbiome over and over and over again, and you're not going to see much difference. It's when you start to see the changes in the conditions or the abiotic and biotic factors that things are going to change, and we know that. So you can't test everything. So it's again the one methodology where if you have primary concerns, say particularly there's microbes of concern, then you would want to know as much as you can about that. But you're not going to be able to test every situation. Sorry, things are. You're not going to be able to test everything, and but it's, you know, more knowledge is always better.
But I liked your first answer, which was simple too, which is it's about, you know, keeping the bad. A lot of it's going to be about keeping the bads down and keeping the goods up, right? So I think
Right.
that's the overall goal.
And the goods are going to probably be the ones keeping the bad ones down.
We're very hopeful for that, but it may be that some of those intermediate bacteria are doing those jobs, and I think those are
That is true.
the puzzle out.
Yeah, it may not be that necessarily the core that is responsible. So you know, you want to know the core, but you also need to know who else is there, because they may be there in lower numbers, but they have a very important job to do, and that's why going to looking at the metatranscriptome or the proteomics, knowing what they're doing is going to be very crucial.
Yes, I'll give you another question, and this was probably be the last question we can get right now, but you had commented before that the hydroponic systems tend to favor bacteria. And of course, we know that there are a lot of mycorrhizal fungi that are important to plant health. So do we know that there are beneficial ones that are in these systems? Have we been able to identify any ones that are beneficial in those hydroponic systems? Have we been able to, or even identify any in the veggie pillows?
We have done the fungal analysis from the veggie pillows, the plants, and the root modules, and there are fungi there. We haven't, I guess, we haven't really drilled down in to see, you know, what, you know, their major roles might be. We've just started into the metatranscriptome work. Hopefully by next year, we'll know a little bit more about that. Because we will be working with some of that tissue.
Great, thanks, Tina. Actually, I'm going to
They
throw
are
one
there.
more question at you, just because it's a very important question, which is one of my colleagues from Europe has said, well, you know, you can't ship seeds across country lines unless they've been sanitized, because you don't want to be moving plant pathogens across country lines. What's the rules for space for the ISS and the moon? Do you have any insight into that?
Well, currently my knowledge of that is that they do need to be sanitized. And for everything that we have sent to stations so far, is that like for the veggie units and stuff, they are sanitized. We have different methods of sanitizing them, and we confirm that prior to planting. And so it's currently we are sanitizing those seeds.
Great, thanks. Well, we're out of time, so I'd like to thank both of our speakers for presenting today, and then just put out a plug for next week's seminar, which will be presented by Annalisa Paul, and there we'll be looking at the cutting-edge science tools really to enable this work for the space science community. So with that, I'd like to thank everybody for attending today's session, and we'll go ahead and close it out. Thank you.
Thank you.
Thank you.